Hello, I'm Robert Costa, national political reporter here at the Washington Post. Joined today and honored to have Sean Parker, a technology disruptor going back to his days with Facebook, now Spotify, and of course now with cancer with the Parker Institute for Cancer Immunotherapy. Sean, thanks so much for being here at the Washington Post. Really appreciate it.
Thanks for having me.
So when you think about what you're doing with cancer, you've given $250 million to the initiative of finding a cure, research. What have you learned as you've just dove into this space?
Well, you know, I think one of the more important learnings were early on. Actually, Stand Up to Cancer was playing a moment ago, and one of the very first contributions I made in the field was around immunotherapy at a time when immunotherapy wasn't a thing. Now there's almost too much buzz and too much hype around it, and there's, you know, we've gone through the first wave of immunotherapy treatments coming to market, the first cell therapies and the first checkpoint inhibitors. Jim Allison, who's one of our researchers, just won the Nobel Prize for medicine, you know, a couple weeks ago.
Affiliated with your work.
Yeah. And he was I think the first person in the field who actually took the time to talk to me and educate me. And I had read his papers going back to the mid '90s, and he had data from 1995, the first patient he ever treated, a melanoma patient who had survived. You know, and this was probably, you know, 10 years ago, something like that, 10, 12 years ago. And so there was plenty of good data that showed that in melanoma checkpoint inhibitors worked, and I just had this strong gut feeling that the field of immunotherapy was going to become the next frontier in cancer treatment, because we'd been going after oncogenes and the oncogenic driver mutations for a long time, trying to find targeted therapies. We'd had some success, as some of you may know, with kinase inhibitors and a variety of other, you know, small molecule drugs. But the idea that the immune system played some sort of role in regulating cancer was not in vogue, and yet the data seemed to contradict this. And you know, I ended up sort of catching that wave at precisely the correct moment. So the big realization was that the immune system does in fact play a role in regulating the early stages of cancer, that all tumors, you know, eventually prior to or concurrent with metastases, develop immunosuppressive capabilities that shut the immune system down, and that if you can block those with drugs or by creating targeted cell therapies, then you can overcome all of the obstacles that cancer sets up. So
what's the next big breakthrough on that front?
So I'm within PICE. You know, you have the group of people who are looking for new IO targets or immunotherapy or immune oncology targets. You know, so we might have another. So anti-PD-1 is on the market, and anti-CTLA-4. Anti-CTLA-4 is called Yervoy. It was developed by Jim Allison, sold by BMS. Then you have Keytruda and Opdivo, and they're anti-PD-1 drugs. These drugs work great in melanoma, and they're a great companion to other cancer treatments, standard of care in other indications, but they're not necessarily curative. They're pretty curative in melanoma. The other modality is cell therapy, and this is my personal kind of bias, and you can't be a specialist in everything, especially when you get into medicine. You find that you know, you very quickly have to sort of narrow your focus. So I've become kind of the cell therapy guru within our group, and I'm pretty much obsessed with T-cell biology. And we have leading that team, Carl June, who built the first and got to market the first T-cell therapy, and it's really extraordinary. So, you know, in the 1990s, we talked a lot about nanotechnology. I don't remember this, if any of you remember this conversation that was taking place in the technology press, like Wired magazine, or I'm sure the Washington Post ran articles on it. And the idea was that you'd have these little nanobots that were roving through your body, like doing various things. And I'm not sure if anybody knew what these things were going to be made of. I think the notion was that they were going to be like little silicone-based robots, and I'm not sure how that would be compatible with your body. But it was a cool idea, and I remember thinking, wow, that's really cool. I'm gonna have robots in my body, like cleaning crap out of my endothelium when I get arteriosclerosis. It's gonna be amazing. And then I remember thinking like, wow, man, those robots could come with lasers and kill cancer. That would be so cool. And then what ended up happening is no less cool. It just sounds more complicated. Sorry.
What's the timeline for this? I mean, some of your friends, like Elon Musk, say because of your work, cancer could be, these kind of technologies could help eradicate cancer in 10 years.
So I am super optimistic. And the reason I'm optimistic about cell therapy in particular is that the way it works is again, sounds complicated, very simple. Take cells out of a patient's body. You modify them currently with a viral vector, which edits the cell. Okay? You modify the cell so that it sees a target that is you can give it a cancer-specific target, you can give it a cell-specific target, but you reprogram that cell so it targets only cancer, or only the cells you want to kill. And then you arm it with various other capabilities. You make it expand better. You potentially you can do any amount of rewiring of the cellular programming of the cell, of a T cell, because we know so much about signaling within T cells. I mean, in truth, we know a tiny fraction of what there is to be known, but we know enough. So we can reprogram T cells, turn them into this. Once we've produced several thousand of them, you can expand them in culture, and you can grow hundreds of millions or billions of these cells. And then you just give the cells back to the patient. The cells kill the cancer, and then they leave your normal cells alone. And that is a.
Is this different than gene editing? Because you've been a huge investor in gene editing.
Well, so you're probably thinking like CRISPR-Cas9, and CRISPR-Cas9 has so many different applications depending on what you're trying to do. I mean, the original hope with CRISPR-Cas9 is that you could fix, remove, maybe do germline gene editing, meaning you're actually modifying an unborn baby, removing a genetic defect, or fixing genetic defects in adult somatic cells in order to restore function, correct various heritable diseases. That's where most of the conversation has been. But that's the as fast as CRISPR-Cas9 is at making gene edits, the fact that it still relies on things like viral vectors that you use to transfect the cell make it really slow. And we've had some major breakthroughs in the last, 90 days.
What's the key one in the last 90 days?
So the two papers published by Pisces researchers. Actually, one of those papers is not yet published. So I can't get into too much detail.
You can give us
the
scoop here at the
But
Washington.
I will give you the. It is coming out soon in Cell, but I'll give you this sort of high level, and anybody sophisticated can fill in the blanks. The high level is that you can use electroporation or whatever technology you want, really, just to push a guide RNA and Cas9 into a cell, and that you can now, for the very first time, make a cut and insert a very large amount of DNA. So this was published in Nature, I don't know, 6 months ago or whenever it came out, and we've shown that you can insert an entirely new T cell receptor, 1,500 up to about 1,500 bases of DNA into a cell without killing it, without harming it or messing it up in any way. And the electroporation is probably the most harmful part of the process, where you literally just shock the cell, and for whatever reason, no one's quite sure. Once you electrocute a cell, you have you can get this CRISPR-Cas9 cassette to go in and make the edit in the correct spot. So what that allows you to do is insert huge amounts of genetic programming into a cell very, very rapidly, so that, let's say you wanted to experiment with mutating a whole variety of different genes, or starting to get into what we used to call the junk part of your DNA. They say 25,000 or so genes in the coding region, and then there's zillions of other genes that apparently don't do anything. Well, it turns out they do a lot. They regulate the expression of genes in the coding region. So this is all sounding very technical. The reality is we can now go in and we can, with incredible precision and control, reprogram the way these cells function. So in essence, they are those little nanobots that we were talking about in the 1990s, but it turns out that they're made of your own cells. They're your own cells reprogrammed. So we can now iterate through with this very rapid process with huge, massively parallel.
How much does it cost, Sean? I mean, if you spent your whole career thinking about accessibility all across all these different.
I mean, I'll give you an example of one experiment that was done by a lead investigator and a pair of grad students in a week. So they effectively knocked out every gene in a T cell. And they only did it in one type of T cell, what's called a CD8 cell, which is an effector cell. It's a killing cell. And they didn't do it in every state of the cell. I mean, generally what you'd want to do is look at a cell after it's killed, or look at a cell when it's exhausted, or look at a cell when it's been suppressed by cancer, and then knock out every gene and see what happens. They did something simpler, which is they knocked out every gene in the cell one by one. That's a lot of gene knockouts. You can imagine how many months or years that would take using normal methods. And then looked at things like how quickly did the cells expand? Did they expand faster or slower? Maybe they looked at a lot of other markers. What came of this was they recapitulated in a week 30 years of work that had been done by the leading immunologists in the world, in the field. People who started their work maybe on HIV in the 80s or even previous to that, and had discovered all of these things like immune checkpoint inhibitors, or immune checkpoints like CTLA-4 and PD-1. These popped up. These genes all popped up, and they were able to find all of this stuff, and you can bet that they found a bunch of new targets that no one had ever heard of. Because when you have an inflection point in the any given industry.
Is this an inflection point right now?
I think that's what makes this so interesting. To your original question, is that there's enough technologies coming together. We all know gene sequencing is happening, but rapidly parallel gene sequencing. Now suddenly we really do have enough data to justify big data and AI. I've been going on panels like this one saying, well, it may be premature to think that Google's going to solve all of our problems in healthcare because they've got a great AI team and tons of computational power. We just don't know enough about the biology. Well, now that you can do massively parallel, totally unbiased screens where you can understand exactly what a gene does, its functional relevance to a particular cell in a particular tissue in a particular individual, whether that individual is healthy or has cancer or whatever, that unlocks this ability to interrogate cells, to learn about their function, learn about the circuitry of the cells, how they're wired, basically. Just having a map of the human genome is pretty damn useless if you don't know what the genes do. And so we're now entering a realm where that discovery process is going to happen much, much faster, and it's not going to require like this 10 or 20-year laborious process of trying to understand some little piece of T-cell biology in order to get your PhD, and then eventually, you become a specialist in that one narrow little area. I think we're going to see a real explosion in terms of how quickly we can move, and not just because of DNA sequencing, or not just because we can use CRISPR-Cas9 to very rapidly test things in a cell to see what they do, but also because you have all these other crazy tools. Like you can look at, you can do RNA-seq and look to see, what proteins.
Who's going to do this? Is Google, Amazon, are they doing enough? Are they being effective?
They're not going to do this stuff because the thing that I had to overcome coming from computer science to biology was that, as complicated as building, back-end infrastructure for Amazon, being able to support that much data, as hard as that is, that's a hard problem. Being able to support 1 billion+ users at Facebook at scale, that's a hard problem. These are only really hard technical problems, but tech people coming from tech to biology so dramatically underestimate the complexity of the human body, and the amount of it's not designed by us. It doesn't work in ways that make sense. and it requires, we have such we maybe know the function of like a half of 1% of genes. So like when if you get your whole genome sequenced, which you can now do for like $1,000 or less, they can't really tell you that much. It's actually incredibly disappointing. Anybody who's paid that $1,000, they're like, well, we can tell you what you're not going to get. no idea what you're going to get. We don't know what the other 99.9% does.
You're talking about tech companies, your work has really sped up the process on cancer. Could Amazon and Google and Silicon Valley get involved in healthcare? Is that going to speed up healthcare?
Well, they are. They are getting involved. I just don't think the innovations that are going to drive this revolution in healthcare and discovery are going to come out of Amazon or Google. And Google has a big group that's focused on this. They're really smart. They're not unsophisticated. They're not naive. But I don't think that's where you're going to see the big breakthroughs happening. Not to say that the ivory tower of academia is like, the be all end all. It's riddled with problems, and it's very slow, and it's incredibly frustrating, and we're all somewhat aware of what.
How are you making sure the right scientists get the funds they need?
Well, we're a different kind of model. I mean, we one of the things that got me to do this, that motivated me, was that I looked at all these charities that were set up that were disease specific. Milken did a great job with the prostate. It's a prostate foundation, and pancreatic has a big one, and there's several colorectal cancer ones. I mean, they're very disease specific, and they basically sprinkle like little grants on a whole bunch of things. It's like a little bit of fairy dust here and there. By and large, the bulk of the research funding is still coming from the NCI, and should be. We need to continue to fund the NIH. This is the plug for funding NIH and NCI.
Will it happen under divided government? You're close to congressional leaders. Do you expect that funding to continue in divided government?
We've done a great job. We did a great job with 21st Century Cures at the end of the Obama administration. That ended up getting fully funded in the GOP-backed funding bill.
What about now?
When we just succeeded in the last budget and continuing to increase funding. The NIH had been down, on an inflation-adjusted basis. They were I don't remember if it was 15, 20%, whatever it was, down from where they should be, had their funding been increased at the rate of inflation. And so we've gotten them back there for the most part. I think more funding is needed. This is important not just in terms of taking care of people and making sure that great research is getting funded. It's also important in terms of American competitiveness in the global economy, because supply and demand will tell you more clearly than anything else where the next several $1 trillion companies are going to get created. And we have a global aging population. Healthcare costs are rising. A larger and larger percentage of all of the income that you make in your life is going to go towards healthcare. So naturally, healthcare, biotech, pharma, everything in that domain, the more that consumes of our GDP.
Have you spoken to President Trump about this?
I've spoken to other people in the White House about it,
Names?
It's a priority.
Any names?
It's a priority. I shouldn't be. I should be, discreet in my conversations.
You don't need to be discreet.
I should be. I mean, I don't need to be. There's no rule that says I have to be, but I should be.
Do you think they share your priorities on cancer? And
I think that if you see it through the lens of both, this is important for lowering healthcare costs. If we're not leaders, if we're not innovating, if we're not at the forefront of CRISPR. And the revolution that's happening there, if we're not pushing the envelope in other areas, we could lose our global leadership advantage
what about his
to
FDA?
China.
What about the Food and Drug Administration under President Trump? Have they been helpful or unhelpful to your causes?
Well, I mean, I think you had Scott on stage earlier, and I'm a huge fan of Scott's, and I think we have the most aggressive FDA in terms of trying to get the process unstuck and get things moving faster. Ultimately, it would be nice to see a world in which we can use more real world data to make decisions about drug approval. That maybe the system for drug approval that we have now, could be tweaked in various ways in order to get drugs to market faster. And certainly for rare diseases, this has been done over the last several decades. Various things for orphan drug indications and things of that nature have happened. Fast track has happened, and certainly for diseases like cancer, the FDA has been pretty fast. I mean, if you have a drug that's at least somewhat better than its predecessors, you'll get, in an indication that's terminal, you can get approval pretty easily. I mean, the problem is that a lot of these drugs only extend your life by 6 months or a year or something like that would be a great outcome in many cases. I'm optimistic that cell therapy, for instance, if you could reprogram a cell to not only recognize tumor, but give it the ability to traffic into the site of the tumor, turn off whatever programming causes that cell to get shut down when it gets near the tumor. When T cells get close to cancer, various weird things happen. We talk a lot about what's called the tumor microenvironment, and it's just this hostile, like apocalyptic terrain that the last soldiers alive in your body have to march through, and they never make it. They're just crap out, and then they almost become like zombie cells. They become energetic, and they start pumping out these things called cytokines, which are immune hormones that cause other cells to shut down. So like every cell that gets anywhere near like a pancreatic cancer cell doesn't make it. But you can reprogram. It's possible to reprogram cells so that they do survive and they do reach the cancer, kill the cancer, and then they turn into memory cells and they hang out. And if a single cancer cell pops up anywhere in the body, it's going to get recognized and it's going to get killed. And that's going to prevent the cancer from ever mutating around this defense system that you've now set up. This is whether this is done through genetically modified T cells that are taken outside the body and re-administered, or if someday we'll be able to do this by just injecting someone with a viral vector or a cancer vaccine. It would be a lot more convenient if you could give this in a normal drug-like fashion rather than having to manufacture cells outside the body. But Carl June and Novartis have proven that they have a great product with a big market, and Kite was sold for however many billion, $5, $6, $7, $8 billion. Another cell therapy company, Juno, similar outcome. So Gilead and Novartis, and Celgene are now all in the game, and they're spending billions of dollars to produce cell therapies for more indications. So we're going to see a lot more of this, and right now it looks like these drugs are going to increase healthcare costs. That's the fear. These are $500,000 treatments, very expensive, a lot of hands on these treatments, very manual process, not super automated, doesn't happen at your bedside. They have to take the cells, send them out, modify them, turn them into a clone army of cancer-killing assassins, freeze them, send them back, unfreeze them, hope they're still alive, give them to you, and then you're like, is it gonna work? What's going to happen? And it, generally speaking, for ALL or really CLL, or ALL and then CLL, you get responses. ALL, you get fantastic responses in patients who failed bone marrow transplants or couldn't find a donor. Mm-hmm. So we're seeing this work. We just what's going to happen is, as more companies enter the space, as this becomes more automated, because these are curative therapies, they don't just extend your life by 6 months, they cure you. So if you can move to a model where these therapies are curative, and you can start to have curative therapies for pancreatic cancer, the burden on the system of constantly treating cancers and then having a remission and then having a relapse and then having a remission and having a relapse, and kind of the endless care that goes into that, and the endless suffering that goes into having friends and family and relatives or yourself dealing with cancer, which seems to always mutate around all of our best targeted therapies. No matter how good we get at precision medicine, the cancer always seems to escape. I think with cell therapy, you have the chance of real durable, stable cures, and the result of that will be costs of these therapies will come down, and then when you're cured, you're cured, right? Like that's a great, the ongoing cost of care is reduced. the hospital visits are reduced. So the long-term effect, I'm optimistic. I'm much more optimistic today than I was 5 years ago, where I was like, oh my God, the sky is falling. We're screwed. All these drugs cost $150,000. Nobody can afford them. What are we going to do?
Right.
And then you start to realize, wait a second, they're coming. The prices are coming down. Competition is coming into the market. Better therapies are coming. Those better therapies, if they really do cure people rather than just extend their lives. I mean, paying a couple $100,000 for a drug that extends your life for 6 months on average is pretty crappy. Paying a couple $100,000 for a drug that cures you is a different story. And then once that works, you can bet everybody's going to build copycat drugs.
And you continue to build and continue to grow with your efforts. But we only have a few minutes left, Sean. Since we're on public policy a little bit, Facebook, have they had the appropriate response, Mark Zuckerberg, to election hacking and to the breach of user data?
This seems to be a theme of panels that I do now, where in the last 3 minutes-ish, somebody starts to ask a question about Facebook.
He's on to reporters' tricks. All right.
I get where you're coming from. I see where this is going.
You're also deflecting the question.
Yeah, yes. So is the response appropriate? I mean, I think Facebook is going through a kind of internal existential crisis. They're having their own kind of. Everybody I know at the company seems to be very, asking themselves difficult questions about Facebook's role in society, Facebook's role in journalism, Facebook's role in whatever. And I think that's healthy, and. It's like you're this little startup. Trying to make it. You're trying to survive. You're trying to beat off all of these big competitors. Google has. I don't know if anybody remembers Google Plus, but they tried to compete with Facebook, and MySpace was the market leader until like 2008, if you can believe it. And then you had various other attacks from other companies along the way. Snapchat at one point posed a really major threat to the company. So you're in like survival mode, and nobody's giving you any flack for that. And then all of a sudden there's this onslaught of concern.
What should it do then, if it's in this existential moment?
I don't think there's necessarily one answer here. I mean, look, marketing communications, better PR is not the answer. You can't design, you shouldn't try to design your product to be, used less, because that's a weird thing for a company to do. That's like, and maybe if you're like a, liquor company or something, or a bar.
Are you saying it's addictive?
We can cut you off.
It's too addictive.
I think as addictive as these products are, it's very hard to imagine designing a product, changing the fundamental way in which the product works, and the product gets, unless you're getting something out of it, psychologically, in terms of social validation, or a feeling of connectedness to other people, or in the past, I've said like a dopamine hit to the brain when you get a like or a comment, and unless you're getting that, you're not necessarily going to contribute content. So that puts Facebook in a tricky position. I mean, I actually think that society will self-regulate to some degree.
Society? What about the government?
Well, the concerns that the government has are not, they're not like these sociological concerns that we talk about, or that so often get talked about, or certainly that I talk about.
Do you think its future could be as a utility?
I don't think so. I think that there's too many other messaging platforms out there. Messaging is so ubiquitous that it's very hard to say, oh well, Facebook is somehow special because it has so many people using it as a message.
Suppose it's special because of what it means for democracy, what it means for the country. You think about the elections.
So you're only ever going to be as strong as your users and the tools you give your users. One idea that has been discussed, probably within Facebook, I haven't talked to anybody at Facebook about it, but certainly elsewhere, is this notion that you could crowdsource, or you could have a group of people, much like with Wikipedia. I mean, there's only 20 plus thousand or so people who are really managing Wikipedia, really actively going after it. And you could imagine a sort of third-party independent organization, because the problem isn't just Facebook. The problem is Google search results and how they're ranked, and how one can use search engine optimization to game those rankings. The problem is anybody with a feed that's algorithmic. How do you deal with that? And there's no metadata, right? So we don't have any kind of unbiased metadata that doesn't have any kind of political bias associated with it, and it's very hard for anybody to trust Google or Facebook or Amazon or whoever to fix their algorithms. So you could imagine a third-party entity that's unbiased, truly unbiased, where behind every Wikipedia page, if you ever you don't want to do this, but if you ever go to the notes section, you can see the warfare that's occurring. I mean, it's like a battle for control of the page. And there are hundreds of people attacking each other, and half of them are trolls and what are called sock puppets, which are fake people, and they're all at war with each other. But there's enough people who hire firms with fake people over here, and enough people who have firms with fake people over here, that they kind of balance each other out.
Does it make sense to have a democracy, a country that has elections that rely on Facebook to provide a lot of voters information, to have sock puppets and trolls?
Oh no, I didn't say on Facebook. I said on some sort of third party. Some sort of.
Well, you compared it to Wikipedia. What else would it be?
Well, Wikipedia is an example of a third party that self-regulates, and I'm sure we've all experienced that it's full of inaccuracies, but it can be corrected. And if you engage with it correctly, or if you engage with Wikipedia editors who are experienced, who have a good track record for editing Wikipedia pages, then you can get a page fixed and you can get information corrected. And Wikipedia, by and large, does a pretty good job of solving a problem that's way more complex than analyzing whether you're dealing with a fact piece or an opinion piece, whether the language in a given article is meant to be emotionally inflammatory, or whether it's unbiased sort of newsroom type language, sort of traditional journalistic language. Those types of things can be solved easily. The question of fact-checking articles is a more complex question, but again, you could see a third party doing that. I guess what I'm saying is it's going to be very difficult for the public and the press to trust that Amazon, Facebook, Google, and others are going to solve this problem.
Final question. You got stuck with me as an interviewer. So 2020, the presidential race. Do you have any, maybe it's President Trump, maybe it's a Democrat, maybe it's an independent. Do you have any candidate who catches your eye? And if so, tell us who. And if not, how do you see it playing out?
It was actually great that I got stuck with a political reporter, because I just made up a whole bunch of crazy science stuff that wasn't true. And
that's true. I wasn't entirely sure what he was saying was spot on every T-cell thing he was saying. But I trust someone would have stood up and called him out if it was really egregious. But I trusted you enough.
Yeah. So the political question was what?
2020. Do you have a candidate who's in mind? And if so, tell us. And if not, how do you think
it's the lightning round? I got an extra minute. Anyway.
It's true.
So.
We should not have the clock when Sean comes back. Kidding.
No, I'm breaking the.
What is it? The fourth wall.
Fourth wall.
Third wall.
Third wall.
You're the Silicon Valley guy. You should know these terms.
Fourth wall.
Fourth wall.
I don't know.
Okay.
Whatever.
2020.
Anyway, so I don't. It's too early for me to have an.
Well, what do you want in a presidential candidate?
I think we want what everybody wants, which is we want stability. We want good governance. We'd like a boring president.
What about Vice President Biden? I'm not saying he's boring, but he's worked on cancer. I'm sure you know him.
Yes. No, we know each other really well. I mean, he's great. And he has a very big heart, and he has a lot of empathy, and he's not boring. He's very engaging. I don't know what he's going to do. I mean, 2 years ago, you know, he would have said absolutely not. You know, I don't know. I don't know. It's hard. I don't want to speculate about who's running, what do I feel about them. It's going to be hard to win a primary at this moment in time without breaking through a huge amount of noise and a huge, you know, we're going to see in the Democratic field something similar to what we saw in the Republican field. And what happens when you have 10 or 15 candidates running is that the kind of mainstream candidates tend to split the vote, and then you end up with the least boring candidate. So, I don't know, maybe one way or another we'll end up with, you know, your job is too fun right now. That's the problem.
You were about to say something good.
It's just,
Don't turn it on me.
Your job is too fun.
What were you about to say?
No, I'm actually
You have
I'm just
something to
thinking it was great back when people used to say like, oh God, who even cares? They're all the same. Just they all are exactly alike. These candidates are so similar. I can't even politics. And now it's like all anyone talks about. You know, I have to pay people. It's like, can you get someone to talk about something else? Like here, we're supposed to be talking about cancer. Look what happened.
We talked about cancer.
Sean Parker, that's all the time we have for today. A reminder to our audience, thank you to Sean Parker. Thank you, I appreciate it. And just wait one second. A reminder, if you want to watch any interviews from today's program or catch up on past programs, you can find that all at WashingtonPostLive.com. Another round of applause for Sean Parker. Thank you.